A very warm welcome to everybody around the globe following this Learning and Memory session at the virtual 2020 OHBM annual meeting. My name is Guillan Fernandez. I'm working at the Dundas Institute in Nijmegen, the Netherlands. I have the privilege to chair this session together with Veronique Bohbot from the Douglas Research Center at McGill University in Montreal, Canada. In the background, you see the colorful glass facade of the Convention Center in Montreal, where the OHBM meeting should have happened. I took this picture when visiting the site while preparing the meeting as a member of the OHBM program committee. Today, I would like to express my gratitude to all those people who have worked so hard to enable a great meeting in Montreal. Be reassured, we will come back as OHBM community. But now we go into the session on learning and memory. The first presenter is Eva Berlot from the Brain and Mind Institute at Western University in Canada.
Hello, everyone. My name is Eva, and I'm interested in how we learn motor skills, such as playing piano. And one important question in brain imaging of learning is where these newly acquired skills are stored or represented in the brain. And today I will tell you how two metrics, repetition suppression and multivariate pattern analysis, can give us very different answers to this question and why we think this might be the case. So one approach to examine where skills are represented in the brain is to measure brain activity, let's say in a specific brain region, while performing a given skill, for instance, a finger tapping sequence. If we're using fMRI, then our measurement channels are voxel, and brain response during skill execution can be characterized as a dot in this voxel space, here simplified by just looking at two voxels. And we can measure brain activation during condition A and condition B. If the two conditions are associated with different or dissimilar activation patterns, then we can conclude that this brain region must represent some feature that distinguishes A from B. And this really lies at the core of pattern dissimilarity techniques. Another way to examine brain representation is by using repetition suppression. In this approach, we first present condition A and then repeat it again. And what is commonly observed is that on average, repetition leads to lower activation than initial presentation of a stimulus. If we want to know whether this effect is stimulus or condition specific, we can first present condition B followed by A. If response to A then is higher than during the repetition, then we can similarly conclude that this brain region must dissociate A from B. So both of these metrics tell us something about what a given brain region represents, but they also differ from one another. Pattern dissimilarity is a slightly more direct way of assessing representation, but repetition suppression can sometimes capture representation on a lower spatial scale. And while they often give similar results, they can also produce very discrepant findings. An example for that comes from the domain of motor sequence learning, where a study using repetition suppression showed that the primary motor cortex represents sequences of finger movements after training. But a study using pattern dissimilarity concluded that this isn't the case. Today, I will first show you that we can replicate the discrepancy between these two findings in a single data set, and then go on to explain why we think this might be the case by delving a bit deeper into the nature of repetition suppression. In our study, we trained participants to execute sequences of 9 finger movements on a piano-like keyboard device, and we trained them on 6 such sequences over a period of 5 weeks, and their performance improved over training. We scanned them both before and after training, and inside the scanner, participants performed trained as well as matched untrained sequences. We used the continuous trial design where sometimes sequences were repeated and sometimes they weren't. And this allowed us to contrast brain activation when a given sequence was novel or presented the first time relative to when it was repeated or executed the second time in a row. And here I'm just plotting the brain activation during the first execution of the sequences, average over all sequences in the first week, now presented on a flattened surface. And to orient you a bit, the two regions I'll be focusing on the most throughout my talk are the primary motor cortex, or M1, which sits anterior to the central sulcus, and the superior parietal lobule, or the SPL, which is superior to the intraparietal sulcus. Relative to the activation on execution 1, execution 2 was associated with lower activation, which can be better appreciated in this difference image, with blue meaning lower activation on repetition, so repetition suppression. The strength of repetition suppression increased with learning, so going from week 1 to 5, in regions that show task-evoked activation. Now, if we look at pattern dissimilarities, we can see that regions that show task-evoked activation also show dissimilar patterns for the different motor sequences. In SPL, both repetition suppression and pattern dissimilarities increased with training, really suggesting that this brain region represents different sequences as more dissimilar from one another after training. In M1, we observed that after training, repetition suppression was stronger for trained than untrained sequences. But in terms of pattern dissimilarities, the two types of sequences did not differ from one another. So essentially, this replicates the previously reported discrepant findings that if we look at repetition suppression, it seems to be the case that M1 represents trained sequences, but in terms of pattern dissimilarities, this doesn't seem to be true. So why are these two different metrics telling us something fundamentally different about whether or not M1 represents trained sequences? To look into this, we've decided to combine the two techniques and look at how dissimilarities change with repetition, so going from execution 1 to 2. And I've told you that activation decreases with repetition. So a simple thing that could happen to representation is that it just scales with activation, meaning that as much as activation decreases, dissimilarities decrease proportional to that. And here I'm just plotting dissimilarities now split by execution, so execution 1 and 2, and the difference between the two. In SPL, we can see that dissimilarities decrease ever so slightly going from execution 1 to 2. And if we compare the similarities observed on execution 2 to what we would expect under scaling, where 0 here means perfect scaling, we can see that SPL's dissimilarities are pretty well accounted for by activation scaling. In M1, we see that dissimilarities drastically reduce going from execution 1 to 2, which can be better seen in this difference plot. So perhaps they decrease more than we would simply expect from activation scaling, meaning that representation really compresses or fatigues with repetition. And indeed, if we quantify this effect, we see that the similarities on second execution in M1 are significantly lower than what we would simply expect under scaling of activation. So we see that representation in SPL and M1 changes differently with repetition. In SPL, we see representation scaling with activation, whereas in M1, we see this collapse of representation, which hints at what M1 represents might be different between executions 1 and 2. And to examine this in more detail, we've looked at all the activation patterns for the different sequences participants performed and tried to characterize them using different features. And we've done that in the pattern component modeling framework. The first feature we looked at was the starting finger of the sequence. So whether two sequences start with the same finger or different fingers. Next, we looked at all fingers. So how many times is a given finger repeated in a specific sequence and how correspondent are different sequences in terms of that. Then whether a sequence was a trained or an untrained sequence and which specific trained or untrained sequence was being executed. And we fit a combination of these models to our observed representation in SPL and M1 across the two executions and tried to see how much variance is accounted for by these different models. In SPL, we saw that most of the variance on the first execution is explained by sequence types, so trained versus untrained, and with specific trained sequences being pressed. Going from execution 1 to 2, the variance explained reduced, but proportionally so for all the different features, meaning that features explaining most of the variance on execution 1 still explain most of the variance on execution 2. In M1, we saw that its representation on the first execution was dominated by the starting finger of the sequence, which is also something that has been previously reported. So this means that the dissimilarities we see amongst the different sequences in M1 on execution 1 are really driven by the different starting fingers rather than sequence identity. Going into execution 2, we saw that this importance of the first finger effect drastically reduced, even more so than for some of the other features. And this likely explains why also we saw this collapse of dissimilarities in M1 on execution 2. So we see that the nature of repetition suppression is quite different between SPL and M1. In SPL, on execution 1, different sequences are represented as distinct from one another. This translates into a strong first finger effect in M1, which perhaps suggests that the code M1 receives from SPL, as well as from premotor regions, primarily prepares the starting finger of the sequence. And then throughout sequence execution, it still receives this input from other higher order areas, telling it which finger is next. Going from execution 1 to 2, activation reduced, but the nature of representation in SPL was quite stable. Whereas in M1, we saw this collapse of the first finger effect, perhaps suggesting that this preparation of the first finger is not as needed the second time around. Going back to the original question of whether or not M1 represents trained sequences, we think the more likely answer is no, because we don't see evidence for it representing sequential context. This doesn't, however, explain why we saw a stronger repetition suppression effect for trained and untrained sequences in M1. So one suggestion is that what really changes with training is that SPL's communication with M1 becomes more efficient. So I've told you how using repetition suppression together with multivariate pattern analysis can tell us something new about motor sequence learning and the underlying circuit. But hopefully you can use this just as an example of how the two techniques can be combined to provide new insights into brain function more generally. And with that, I'd like to conclude, thank my lab, the Dillan lab, for all the help and support, and thank you for your attention.
Good morning, I'm Jiwoong Park from Sungkyunkwan University. Today, I'll be talking about neural correlate of individual differences in story understanding. Understanding stories is arguably one of the most unique human behavior. The ability to understand stories is needed not only for reading books or watching movies, but also for making sense of our own memory and others' behavior. Interestingly, even when everyone encountered the same story, the interpretations of its meaning may differ across individuals. For instance, the animated movie Alice in Wonderland opens with a talking white rabbit who is going somewhere while Alice tries to follow him. Even in this simple narrative, there can be various interpretations. Some may wonder how the rabbit can speak, while others may focus on where the rabbit is going. Recently, researchers have started to adopt naturalistic experimental paradigm by using narrative movies. Previous studies found the neural representation that were involved in the process of experiencing narrative content. For example, using fMRI, Chen and colleagues found that neural pattern induced by each scene of the movie was shared across individuals. It was also shown that event structures of the movie were represented by the brain region with a long temporal integration window that can be used to integrate narrative information. Unlike those studies on the common neural processes involved in story understanding, some researchers have focused on a neural representation that reflect individual differences in processing narrative. For example, recent work by Bajatram and colleagues, where subjects were divided into two groups depending on their thinking style, showed differences in the pattern of brain activation between analytic and holistic thinker while they watch the movie. In our study, using fMRI and natural language processing, we aimed to find the neural signature that underlie such individual differences in story understanding. Our research question is, first, what are the dynamic changes in the brain state that are correlated with narrative story understanding? And second, what are the neural signatures that differentiate individuals who understand the story well from those who did not? To examine this question, we conducted an fMRI experiment where participants watched a scrambled version of a movie and later recounted the contents they viewed. We scrambled the movie in its temporal sequence to increase the difficulty level of narrative comprehension to maximize individual differences. Here, our goal is to link brain state dynamics during movie watching to an individual's ability to understand the story. To assess each individual's story understanding ability, first, we had the original movie annotated by multiple annotators to make a ground truth data to compare with the individual recall data. Using natural language processing and topic modeling, we transferred the narrative information to topic vectors, then quantified the semantic contents of individuals' recall in comparison with the reference topic vectors. We assessed the story understanding ability of each individual using the similarity between their recall and the original movie annotation. In the right figure, the x-axis indicates each sentence of individual recall, and the y-axis represents the actual sentences of the original movie. For each sentence in individual recall, we calculate each similarity to the reference sentence. If a subject recount the original story well, you can see a clear diagonal, as shown in the data of subject 3. From this similarity matrix, we computed 2 quantitative measures to assess the quality of individuals' recall: the total amount of information covered and the order of information. First, we quantified how much semantic information from the original story was covered in each recall. For each time point in the original movie, we averaged the matched information across all recalled sentences and made a distribution of the semantic information covered in the recall across the duration of the movie. Then we compared this distribution of the recall with the distribution of the reference. The differences between these 2 distributions indicate the amount of information the recall has. The smaller this value is, the larger amount of information was included in that recall. Then, to take into account the order of information in the original movie, we computed another measure to indicate how closely the individual recall reconstructed the order of event in the original movie. If the subject recounted the original story in the correct order, we could expect a strong orderly diagonal on this matrix. We estimated the correctness of temporal order by calculating the rank correlation between reference and recalled sentence. Finally, we normalized 2 measures and combined them into one story understanding score, which are plotted on this figure. Each point here represents a subject. The x-axis indicates the amount of information in an individual recall, and the y-axis indicates the correctness of the order of information. The individual's story understanding score was computed as each distance from the reference point based on the annotation. The darker the color of each point is, the higher that subject's understanding score is. A subject in the upper left corner with a high story understanding score shows good record performance in terms of both amount of information recorded and its correct temporal order. In comparison, a subject in the upper right corner record the information in the correct order, but the amount of information was low. A subject in lower right corner show poor record performance in terms of both amount of information and its temporal order. For the next step, we aimed to identify the brain state that was correlated with this individual story understanding performance. Because the story understanding is dynamic process, to find the dynamic brain state that reflects the cognitive state, we conducted a separate behavior experiment where changes in cognitive state were measured. In this behavior experiment, an independent group of subjects rate their subjective level of understanding while watching the same scrambled movie. Subjects reported whenever they thought they had understood the story. The responses of all subjects were aggregated with sliding window, and the blue line on this figure represents the moment by moment subjective understanding level while watching the movie. Next, we focused on identifying brain state that varied with this measure of story understanding. To capture the changes in the brain state, we used the functional neuroimaging data acquired during movie watching. This method was adopted from recent work by Chung and colleagues. First, in each ROI, parcellated based on atlas by Fan and colleagues, we extracted the time series of each voxel, then reduced its feature dimensionality using nonlinear dimensionality reduction technique. Then we detected a moment of transition, a discontiguous jumping point on the neural trajectory in a 2D embedding space, which is indicated by blue peaks. We characterize the dynamic brain state based on this pattern of transition. A red line here represents the smoothed transition pattern. So we correlate the brain state transition in each individual with the cognitive state changes during movie watching. We assume that if the correlation between the brain state and cognitive state was high, it would indicate adaptive reconfiguration of functional network, which is required to appreciate the story. So as a final step, we correlate the degree of synchrony between the brain and cognitive state dynamics with individual story understanding score. In this example ROI, individuals' brain state were more synchronized with the cognitive state in the high understanding subject compared to the low understanding subject. Using this method, we examined whether there were specific brain regions that correlate with the individual differences in story understanding. We found that most ROIs shows a negative correlation with story understanding score. This result suggests that this pattern of brain and cognitive state synchrony is associated with story understanding ability. On the large-scale functional network level, we found the degree of the brain and cognitive state synchrony in the dorsal attention network was significantly correlated with an individual ability to understand the story. Taken together in this study, using fMRI and natural language processing, we examined the dynamic neural signature that underlie individual differences in story understanding. Our results indicate that during the process of story understanding, high understanding subjects show a high degree of brain to cognitive state synchrony in the attentional network. These results suggest that dynamic coupling between brain and cognitive state may play a critical role in our story understanding ability.
A gradient from long-term memory to novel cognition transitions through default mode and multiple demand network. Human cognition flexibly guides decision-making in familiar and novel situations. Traditionally, these different aspects of cognition have been ascribed to different neural systems. When the knowledge required by a task is readily available within long-term memory, gradients of default mode are assumed to play a role. In the absence of long-term knowledge, the multiple decisions are supported by multiple demand network. However, human cognition is a continuum, not a dichotomy. Most scenarios locate different positions on this continuum. Contemporary accounts of cortical organization suggest the intrinsic connectivity and functional organization may change gradually and systematically along the cortical surface. The following is what they did. They constructed a whole brain functional connectivity matrix. Take the connectivity graph here. If the functional connectivity pattern of two nodes are similar, they would be close to each other. Then they apply a dimension reduction technique to capture the maximum variance along several dimensions in this connectivity matrix. And then they project its components. Here shows two gradients they have found. And two gradients would occupy similar positions along the gradient if they resemble each other with respect to the connectivity component of interest. The principal gradient, gradient 1, extends between the sensory motor regions and the transmodal regions. Our study mainly focused on the first gradient, and the gradient also captures the spatial layout of the networks found by Yale. Principal gradient values of these networks show gradual transition, and the gradual transition appears in multiple zones. This principal connectivity gradient offers a view of neural organization, so it shows there's a gradual transition across the systems, and this gradual transition appears in multiple regions. Motivated by this view, we wondered, does the cortical connectivity gradient reflect how decisions change in a graded fashion as the support from long-term knowledge changes? We would like to answer these questions. If we manipulate long-term semantic similarity parametrically, does the functional recruitment change gradually? Is there a spatial correspondence between the effect of long-term semantic similarity and the principal connectivity gradient? Does the correspondence between the task gradient and connectivity gradient appear in multiple regions? Can we explain the spatial layout of semantic networks in terms of their positions along the task gradient? We created an experimental paradigm in which participants decided whether concepts shared a specific feature, such as color. We manipulate their overall semantic similarity parametrically, creating a spectrum of global similarity. For some trials, they shared many features, such as color, strawberry, and cherry. For some trials, only the target relevant feature was shared, such as color, tomato, lipstick. We have two conditions. In one condition, nearly the probe and target share task relevant feature. In another condition, probe and target don't share target relevant feature. We examined the parametric effect of global similarity by including it as a parametric regressor. For matching trials, global similarity ratings is higher, the accuracy is higher, and response is faster. For mismatching trials, global similarity rating is higher, the accuracy is lower, and response is slower. Global semantic similarity rating has different impacts on behavioral performance for these two conditions. Consequently, we can compare them to establish whether the functional change along the task gradient reflects the global semantic overlap or task difficulty. Global semantic overlap would produce parallel task gradient. Difficulty would produce opposite task gradient. We model the parametric effect of global feature similarity. This figure shows the estimated effect of the parametric manipulation of global feature overlap. Default mode gradients show positive effect of this variable, that is, a stronger bold response when items share more features. Multiple demand gradients show negative effect of this variable, that is, a stronger bold response when items share less features. To establish whether there's correspondence between the task gradient and the connectivity gradient, we calculate the correlation of these two maps, and we found a significant correlation. And since we used a semantic mask, we wondered whether the correlation would be stronger if we only included regions that are involved in semantic cognition. We defined a semantic mask. We found the correlation is stronger. This suggests the correspondence between the task gradient and the connectivity gradient is to some extent specific to brain regions relevant to semantic cognition. To establish whether functional change along the task gradient reflects the global semantic overlap or the task difficulty, we repeated the same analysis for mismatching trials, and we found a similar pattern rather than opposite pattern, suggesting this effect reflects global semantic overlap. And now we answered two questions. If we manipulate long-term semantic similarity parametrically, the functional recruitment changes gradually, and there's spatial correspondence between the effect of long-term semantic similarity and the principal connectivity gradient, which cannot be explained by task difficulty. We wondered whether the correspondence between the task gradient and the connectivity gradient appear in multiple zones. We anatomically defined these regions: left lateral frontal, left medial frontal, left lateral temporal, and left lateral parietal. We found a significant correlation between this task gradient and the connectivity gradient. To establish if the response to the task gradient systematically changed along the connectivity gradient, we assigned voxels into 20 beams according to their values on the connectivity gradient. We extracted the parameter estimate of each beam, corresponding to the effect of global semantic similarity. We examined whether there's a linear change across these beams, and we found a linear change. And this indicates there's systematic change along the connectivity gradient. The transmodal regions show the positive effect, and the unimodal regions show the negative effect. To examine whether the similar pattern would appear in mismatching trials, we repeated the same analysis and found a similar pattern. This argues against the difficulty hypothesis again. We wondered whether we can explain the spatial layout of semantic networks in terms of their positions along the task gradient. We compared the location of semantic control regions with default mode and multiple demand, as defined by the localizer tasks. We used the contrast of easy versus hard of a spatial working memory task to define default mode. The contrast of hard versus easy of spatial working memory to define multiple demand. The contrast of hard versus easy of semantic task to define semantic control regions. We found the semantic control regions indicated by the green color is in the middle of default mode and multiple demand, especially the lateral temporal cortex. Here's a summary of our findings. The brain's response to the task gradient varied systematically along the connectivity gradient, with the strongest response in default mode when the probe and target items were highly overlapping. This graded functional change appears in multiple regions, and this effect cannot be explained by task difficulty. And this principal gradient captures the layout of the semantic networks, providing an organizational principle of networks involved in semantic cognition. Here's the conclusion. Cognition is organized in the brain in a graded fashion. The dichotomy between default mode and multiple demand might represent two ends of a gradient. Semantic tasks recruit both networks and the territory in between, depending on the match between the input and long-term memory. I'm very grateful to my supervisors and my collaborators. And thank you.
All right. Hello, OHBM. I'm Dillan Newbold, and I'm an MD PhD student at Washington University, working with Nico Dosenbach. Our lab is interested in mapping individual human brains using densely sampled resting state fMRI. My project expands on our densely sampled approach by using daily 30-minute scans to track plasticity in individual participants during 2 weeks of limb constraint. So this project was possible.
Hey, real quick.
Uh-huh.
I'm gonna just restart you right now because the audio got messed up a little bit. So if you want to just back it up a little, 15 seconds, perfect. All right, 5, 4, 3, 2.
Hello, OHBM. I'm Dillan Newbold, and I'm an MD PhD student at Washington University, working with Nico Dosenbach. Our lab is interested in mapping individual human brains using densely sampled resting state fMRI. My project builds on this densely sampling approach by acquiring 30-minute scans every day to track plasticity in individual participants during 2 weeks of limb constraint. This project was possible because 3 highly motivated and slightly crazy members of our lab volunteered to get scanned every day for 42 to 64 consecutive days. They did 30 minutes of resting state fMRI every night, 16 minutes of task fMRI, 18 minutes of diffusion MRI, periodic strength and fine motor testing, and constant behavioral monitoring using accelerometers worn on both wrists. Every component of this study was a huge commitment for our participants, and I still haven't mentioned the biggest thing. Every participant wore a cast covering their entire dominant arm for 2 weeks. This design allowed us to induce disuse, specifically in the brain circuits controlling their casted arm, and track daily time courses of plasticity during disuse and recovery. With all the data we collected, we were able to acquire very precise measurements. So here I'm plotting out functional connectivity measured between the left and right motor cortex every day prior to casting. So every dot represents one 30-minute scan. You can see that we see a very consistent measurement of about 0.7. This fits with our prior work showing that functional connectivity is highly stable from day to day when enough data is acquired. There's a popular hypothesis that functional connectivity is normally maintained in a Hebbian-like manner by co-activation of brain regions during behavior. We believed that blocking use of one limb would decrease co-activation of the left and right motor cortex, and we hoped that 2 weeks of limb constraint might be just enough to drive a measurable change in connectivity between these regions. Well, that's sort of what we found. This measurement was acquired after just 12 hours of casting, and this was 1 day later. Throughout the cast period, functional connectivity continued to decline, and then returned to baseline within 2 days after cast removal. Although there were some scans up to a month later when we still saw lower than normal functional connectivity. The time course of these effects was much faster than we had anticipated. It appears that functional connectivity is determined largely by a person's most recent experiences and behaviors, although there may be some effect of experiences that last up to a month or more. So we replicated these results very cleanly in 3 participants. and found absolutely no effects in neighboring motor regions, which we tested as negative controls. All of these results, and another very exciting finding that I don't have time to describe here, are described in our new paper that will be coming out in Neuron on June 16th. For me, that's in the future, but if you're watching this during OHBM, this paper should be available online right now. So please look for it. Today, I want to spend most of my time talking about something that I've been doing more recently with this same data. So we wanted to look beyond the motor system to understand how casting changed functional connectivity throughout the brain. To do this, we began by examining seed maps showing functional connectivity between the left motor cortex, the disused region, and the rest of the cerebral cortex. You can see that this region at baseline shows strong connectivity with the rest of the somatomotor cortex, especially the homotopic upper extremity region in the opposite hemisphere. During casting, we observed a dramatically different pattern. And then after casting, we observed a pattern that was very similar to beforehand. To examine these changes in more detail, we can look at subtraction maps that show changes in functional connectivity during casting. To simplify this image, I'm showing these maps on a set of cortical parcels. These parcels are specific to this individual, and had actually been previously generated and published as part of our lab's prior Midnight Scan Club experiment, which is described in Gordon et al. In Neuron in 2017. You can see on this parcellation that we saw strong decreases in connectivity with the rest of the somatomotor cortex and increases in connectivity with several other cortical regions. Changes during recovery very closely complemented the changes that we observed during casting, suggesting that all of these effects reversed quickly after cast removal. We can understand these changes in even more detail if we examine them in network space. So resting state data tell us a lot about the organization of the brain. Brain regions that are located in distinct anatomical locations can have closely related functions. And we can appreciate that functional organization using visualizations like this spring-embedded graph. This graph is based on this participant's connectivity patterns prior to casting. And you can see that brain regions cluster into several different communities. These communities correspond to well-known functional networks. And for this participant, these networks had been previously identified and published as part of our Midnight Scan Club experiment, again described in Gordon et al. In 2017. If we examine the changes in connectivity during casting in this way, we can see that decreases in connectivity fall into three clusters. These correspond to the upper extremity, lower extremity, and face regions of the somatomotor cortex. Increases in connectivity were even more focal in network space. Although many different anatomical regions showed increased connectivity with the disused motor cortex, all of these regions fell into a single cluster in network space. If we compare this to the network map that we published previously, you find that this cluster corresponds to one functional network known as the cingulo-opercular network. This is a network that our lab has studied for years. It includes regions of the dorsal anterior cingulate cortex, angular gyrus, and anterior operculum. It has roles in action selection and maintenance of goal-driven behavior. We can quantify the specificity of these increases in the cingulo-opercular network by examining the top 5% of parcels showing increased connectivity with the disused motor cortex. We see that these parcels fall overwhelmingly in the cingulo-opercular network. This result was true across all three of our participants and across a wide range of thresholds. So all of the analyses that I've shown you so far have focused on changes involving the disused motor cortex. But it turns out that these are essentially all of the effects in the entire brain. So this is a matrix showing all changes in connectivity between all cortical parcels and a set of cerebellar segments generated in a similar manner. Matrices like this can be a bit overwhelming, but what you should take away is that most of the matrix is empty and that almost all of the large effects fall within the somatomotor and cingulo-opercular networks. To make this easier to appreciate, we can take the 50 largest changes and show them in anatomical space. So on the right, this is essentially a summary of all the effects that I've shown you so far, though it was generated by looking at all changes between all cortical parcels. You can see decreases in connectivity between the disused motor cortex and the rest of the somatomotor cortex, and increases in connectivity between the disused motor cortex and the cingulopercular network. So we are currently writing up a manuscript describing these changes and what they tell us about the cingulopercular network and its roles in action, selection, and plasticity. Okay, I'd like to thank all of the members of the Dosenbach Lab and our collaborators. I especially want to point out Cole Sider, Annie Jung, David Montez, Deanna Barch, and Ryan Rout, because they're all presenting at this year's OHBM. To find out more, please visit my poster and look for our paper. If you email me, I will send it to you. If you want to know more about precision neuroimaging, visit our lab website at dosenbachlab.wasl.edu. We'll be sharing our data on Open Neuro, and all of my code is available on GitLab right now. If you want to find these links, you can find them all pinned to the top of my Twitter page. So please follow me and my lab to find out more. With that, thank you for listening, and I would love to take your questions in about a month.
Okay, hello everyone. Today I'm going to present my work on striatal GABA, motor learning and stress. This work was done in the context of my PhD, which I just completed at the university, at the KU Leuven University in Belgium. Memory in humans has historically been classified in the declarative memory system supported by the hippocampus and the procedural memory system associated with cerebellar striatal structures. The latter supports our elaborate repertoire of motor skills that we use on a daily basis. Importantly, in the last decade, more integrative models have been developed, and there is now considerable evidence that the hippocampus interacts with the striatal system during motor learning. Indeed, previous research has shown that the hippocampus is also involved in motor memory processes, and in particular in motor sequence learning and memory. Crucially, it is suggested that optimal memory formation and consolidation requires a balanced recruitment of corticohippocampal and corticostriatal networks during initial learning. However, it remains unknown whether modulating this balance during memory acquisition influences motor learning and long-term retention. One of the factors that has been found to challenge the balance between memory systems is experimentally induced stress. It has been found to boost striatal activity and to reduce activity in the hippocampal system during both declarative and procedural non-motor learning. Our recent work showed that stress also disrupts the balance between memory systems when induced before motor sequence learning. More specifically, we found that stress favored the recruitment of motor cortical areas and results in the gradual disengagement of hippocampal cortical networks during learning. However, all these previous studies have largely focused on bold responses, and no study, to our knowledge, has examined the effects of stress and motor sequence learning on striatal and hippocampal neurochemistry. Especially with respect to motor learning, this knowledge gap is surprising, as previous evidence provides a link between motor learning-related plasticity and GABA, the main inhibitory neurotransmitter in the brain. In the current study, we used experimentally induced stress to modulate the balance between hippocampal and striatal systems during motor sequence learning. More specifically, we investigated whether stress modulates the balance between these systems beyond the level of bold responses, and therefore combined functional MRI with MR spectroscopy of GABA to assess changes in hippocampal and striatal GABA levels. 55 young healthy participants were exposed to a stressful or non-stressful control intervention before being trained on a bimanual motor sequence task in the MR scanner. As a stress induction methods, we used a socially evaluated cold pressure test, or SECPT, which induces stress due to a combination of pain and social evaluation. During the motor sequence learning task, participants learned an 8-element sequence by repeated practice. GABA levels were assessed using MRS prior to the intervention, as well as at the end of motor sequence learning. Today, I will focus on whether stress and learning modulates GABA in our regions of interest, and whether stress or learning-related modulations in GABA link to changes in BOLD. We used MRS, and more specifically the megapress sequence, to assess local GABA levels in the left striatum and the left hippocampus. The striatal voxel was centered on the putamen in the coronal and axial views, and as a result, only part of the caudate was included. The hippocampal voxel was centered on the left hippocampus. What you see here is the consistency of voxel placements across participants and across sessions. Unfortunately, it has been difficult to obtain a good MRS signal for the hippocampus, and due to issues with data quality for this region, I will only present the results related to striatal GABA. On the right, you can see the average in vivo spectrum for the striatal GABA, for the striatum, and the GABA peak can be seen at 3 ppm. Before moving to the neuroimaging results, it's important to show that the intervention triggered significant physiological responses. First, we measured cardiovascular responses before, during, and after the intervention. As you can see here, heart rate, systolic, and diastolic blood pressure increased in the stress group during the intervention. The stress group is depicted in red. In addition, we also assessed cortisol concentration. Cortisol is the principal human stress hormone, and our results show that our intervention triggered a significant increase in cortisol in the stress group, and crucially, cortisol was elevated at the start or reached the peak at the start of the fMRI training session and remained significantly elevated until the MRS post-scan, until the end of the MRS post-scan. With respect to neuroimaging, I will first focus on the results related to MRS. As a reminder, GABA was assessed prior to the intervention as well as at the end of the fMRI training session. Based on evidence that GABA in motor cortical regions decreases over motor learning, and these regions have massive glutamatergic projections to the striatum, we expected a similar increase in the striatum over learning. We also hypothesized that stress would further decrease GABA levels. Unexpectedly, however, we found no differences between groups post-training, and neither was there a change in GABA from pre to post. Next, we investigated whether stress modulated the dynamics of hippocampal and striatal responses during motor sequence learning. What we see is that while stress did not influence the dynamics of striatal responses, as you can see here, responses increased as a function of practice in both groups. Stress did modulate activity in the hippocampus. More specifically, our results show that the right hippocampal activity decreased in proportion to performance in the stress group, and this significantly more than in controls. Lastly, to look at the link between these BOLD responses during training and striatal GABA, we ran an fMRI regression in which GABA was included as covariate. Interestingly, our regression with GABA post showed that individual differences in the stress-induced decrease in hippocampal activity, indicated on the y-axis, were negatively related to striatal GABA post-training, presented on the x-axis. So, in other words, the more hippocampal activity decreased during learning, the higher the GABA levels at the end of the learning session. Our results suggest that stress modulates the interaction between the hippocampal and striatal systems across BOLD and GABA signals. This relationship might seem contradictory at first, as in parallel to the decrease in hippocampal BOLD, we know that striatal activity increases, and therefore it was expected to be linked to lower inhibition. We think that the opposite relationship might in part be explained by the size of the voxel. To obtain sufficient signal, you need to use a relatively large voxel, and as a result, not only the caudate and the putamen, but also the pallidum was included. What follows next is a simplified schematic representation of the potential consequences of motor sequence learning under stress on GABA and the striatal voxel. In green, I represent glutamergic or excitatory projections, and in red, inhibitory or GABAergic projections. Based on physiology, we know that the striatum receives glutamergic projections from the motor cortex and has massive GABAergic projections to both the internal and the external segments of the globus pallidus. For completeness, I also present the thalamus and the subthalamic nucleus here, as the pallidum receives its main glutamergic input from these structures and also has GABAergic projections towards these structures. However, I will focus on what happens in the regions included in the MRS voxel. We speculate that under stress, the shift from hippocampal to motor cortical networks, I'm referring to the boost in motor cortical regions described at the beginning of the talk, might have increased glutamergic input onto the striatum and therefore increased GABA input to the pallidum. We suggest that the GABA level in the voxel might not reflect striatal inhibition per se, but might detect the massive GABAergic input from the striatum to the pallidum. Although this interpretation is highly speculative, it might explain why larger decreases in hippocampal bolt were related to higher striatal GABA. So, in conclusion, in the current study, we investigated the effect of motor learning and stress on hippocampal and striatal networks beyond the level of bolt. We found evidence that stress-induced modulation of hippocampal bolt responses during motor learning is related to inhibition in the striatal system at the end of learning. And hereby we provide the first evidence for an interaction between these systems across GABA and bolt signals. I would like to thank my team, in particular Brad and Geneviève, and their collaborators for their support with the project. And I would like to thank you all for attending my talk. If you would like to discuss more, I will present my poster on July 2nd between 8 and 9 a.m. Thank you.